Good morning, students. In previous classes in 12th biology, various topics are discussed, and the chapter 5, principles of inheritance. In this chapter we have learned various topics like monohybrid class, dihybrid class, incomplete dominance and codominance, and the chromosomal theory of inheritance. Today, what is the difference between genes and pairs according to the chromosomal theory? Because normally gene, you know, the segment of DNA which conducting the information, which inheriting the information from one generation to next generation. And chromosomes, we know the chromosomes is a structure present inside the nucleus, inside the chromatin reticulum. Normally chromatin reticulum only, it's slowly converted into chromosomes. So I draw the diagram of chromosomes and the genes. Listen. For example, gene means is nothing but a segment of DNA. Normally we know the DNA which present inside the chromosome. So actually gene is nothing but the segment of DNA. A part of DNA is known as gene, which consists of information like nucleotides and adenine, thymine, cytosine, guanine, like nitrogen bases are present. So gene is nothing but is a segment of DNA present inside the nucleus. And its chromosome, we know the chromosome is a structure present inside the nucleus only. During cell division stage, the chromatin reticulum only slowly condensed into chromosomes. So gene, we know the segment of DNA, and chromosome, we know the structure present inside the nucleus, which is actually a chromatin network. During cell division stage, it slowly condensed into chromosomes. So normally genes also occurs in pairs, chromosomes also occurs in pairs. Normally we have various genes, around 30,000 genes are present in a human cell, human diploid cell. Even chromosomes also occurs in pairs. So human contain 23 pairs of chromosome, that is now 46. So gene also occurs in pairs, which according to the Mendel's view, example, capital T controlling the tallness, even another one capital T also which controlling the tallness. So genes also occurs in pairs in a cell, in a chromosome, and chromosomes also occurs in pairs. Normally we know the chromosome occurs in pairs because the human contain 23 pairs of chromosomes. Then gene also, for example, listen, this is actually chromosome. This is actually homologous chromosome. Similar structured chromosome is known as homologous chromosome. Here one gene are present. This is actually one gene. Here also genes also present, the same gene, the two versions of genes. So the genes are present inside the chromosome only. This is actually a segment of DNA. DNA actually present inside the chromosome. So gene also occurs in pair in a homologous chromosome, the same version of two genes. One chromosome during gamete formation move into the one gamete. Here another chromosome move into the another gamete. For example, human male producing two types of gametes. One gamete with X chromosome and another gamete contain Y chromosome. So in X chromosome also same genes are present, even Y chromosome also genes are present. Even XX also in female also. So genes also occurs in pair. Even chromosomes, we know the homologous chromosomes, the 23 pairs of chromosomes also occurs in pairs. So during gamete formation, segregate at the time of gamete formation such that only one of each pair is transmitted to the gamete. Segregation means separation of gene during gamete formation. For example, we have two genes in same controlling the same characters, means one gene move into the one gamete, in the same version another gene move into the another one gamete. So during gamete formation the genes also segregate, and one of the each pair is moving to the one gamete. For example, human male means the X chromosome genes are present in one gamete, in another one gene are present in Y chromosomes gamete. Okay, so even chromosomes also segregate at the gamete formation, and only one of the each pair is transmitted to the gamete. Normally this idea we know, because human gamete, how many chromosomes are present in human gamete? 23 chromosomes are present, we know. In 23 chromosomes the gamete contain 23 chromosome, but in parent cell contain 46 chromosome. Normally the cell contain 46 chromosome. During gamete formation the gamete only contain 23. Okay, so only half the number of chromosomes only segregate and move into the gametes. So then next one, independent pairs segregate independently of each pair. So independent gametes, independent genes means or separate genes which controlling the different characters. The independent genes or independent pairs segregate independently of each other, means the genes are independently separating and move into the gamete and control the particular character. Even chromosomes also, one pair segregates independently with the other pair. One pair of chromosome is segregating independently. It's actually we know that is due to independent assortment law in Mendel's mono-dihybrid case. So one pair segregated independently with another pair. This is actually a simple difference between gene and chromosome. So normally in the chromosomal theory, the simple explanation about genes and chromosomes, how the difference between genes and chromosomes. Okay, yes. Then Mendel only formulated principles of inheritance. In these principles of inheritance, law of dominance, law of segregation and law of independent assortment. Then next to the Mendel, John Mendel, various scientists are discovering chromosomes and other structures present inside the nucleus. The next to Mendel, and the Sutton and Boveri, Walter Sutton and Theodore Boveri, there are two scientists which researched, which developed their small microscopes. With the use of microscope, he analyzing the chromosomes movement in mitosis and chromosomes movement in meiosis, and during gamete formation how chromosomes are segregated. Normally you know human contain 46 chromosomes. In 46 during gamete formation 23 chromosomes segregate from each other and 23 move into the one gamete and another 23 move into the another gamete. The same way the Walter Sutton and Boveri, he observed a chromosomal movement during meiosis division. So with the use of knowledge, with the use of chromosomes and with the use of Mendelian principles, and the Sutton and Boveri and our colleague scientists formulated a chromosomal theory of inheritance. Chromosomal theory of inheritance is nothing but same like Mendel's theory, but in Mendel's words, factors only controlling the genes, factors only controlling the characters, not a gene, factors only controlling the characters, inheriting the character from one generation to next generation according to the Mendel's theory. But in chromosomal theory of inheritance, chromosomes only has the information. Chromosomes only conducting the inheriting the character from one generation to next generation. Only the chromosomal theory. Chromosomal theory simply explains the structure present inside nucleus is chromosomes. It is a colored structure. Chromosome only contain information for the particular character. Chromosomes only inheriting the character from one generation to the next generation. So actually chromosomes only as a genetic material. In according to the Sutton and Boveri view, chromosome only inheriting the characters. But in future, genes, DNA are developed, and various scientists are involved in the DNA and gene isolation. But in Sutton and Boveri, according to the two scientists' view, chromosomes only has the information for making the protein or making the expressing the characters. So chromosomes only inheriting the character from one generation to next generation. That is actually chromosomal theory of inheritance. The particular chromosomal theory of inheritance is verified by with the use of T.H. Morgan, Thomas Hunt Morgan. T.H. Morgan is a scientist. He developed various techniques in mutation. Mutation, we know, sudden changes in the chromosomes, sudden changes in the gene, sudden changes in the DNA. Due to this random change, the different characters are expressed in the progeny. But in Darwin theory, in next lesson, in the evolution lesson, Darwin actually explains evolution is a slow process. Slow changes only occurs in the body of the organism. Due to this changes only, the organism slowly develops into a new species. But it's very long time. It takes very long time. But in according to the Thomas Hunt Morgan, T.H. Morgan, due to sudden mutation, sudden changes in the chromosome, sudden changes in the DNA, sudden changes in the gene, totally the new characters are expressed in this new species. So sudden changes is nothing but that is mutation in a chromosome, in a DNA or in a gene. So how the T.H. Morgan is experimentally proved the chromosomal theory. This is nothing but the experimental verification of the chromosomal theory of inheritance is done by Thomas Hunt Morgan, T.H. Morgan. We take small fruit flies, that is Drosophila melanogaster. In our board examination also, maybe NEET examination also, the question may ask. They give a scientific name of common fruit fly. It's nothing but Drosophila melanogaster. It's a common fruit fly. It's nothing but during May season or a small, large size house fly may come in your home. So the large size house fly during a particular in the mango season is nothing but fruit fly. Is actually Drosophila melanogaster. Okay, it contains various chromosomes and various variations. With the use of Drosophila melanogaster, he proved that chromosomal theory of inheritance. Why the T.H. Morgan taking a Drosophila melanogaster for the experimental organism? Because the Drosophila melanogaster is grow in a simple synthetic medium. Simple synthetic means with the use of simple chemicals, with the use of simple nutrient in the laboratory, we cultivate we develop the fruit fly, that is Drosophila melanogaster. So in an easy experiment, without any advanced technology, without any hybridization technology, with a simple synthetic medium, simple food is enough for producing the fruit flies. It's actually one advantage for selecting the fruit fly. Then another one character for selecting the fruit fly for this experiment, life cycle of the particular Drosophila melanogaster only 2 weeks. Within 2 weeks, it produces the next generation. So in 1 month, we research a 4-generation organism. We multiply 4 generation within 1 month. Within 1 month, 2 generations are produced, not 4 generations, because 2 weeks in a lifespan, and 4 weeks. After 4 weeks, 2 generations are produced. So easily we observe the changes in the generation, how the changes occurs, how the variations occurs, how chromosomes which inheriting the next generation, how due to this variation, due to this mutation, how changes occurs in the progeny. With the use of 2 weeks, we observe the changes in the result of the research. So the T.H. Morgan taking Drosophila because the life cycle is within 2 weeks it produces the next generation. The next point, for selecting the fruit fly for his research, within a single mate is nothing but reproduction. With a single reproduction, male and female Drosophila. Normally male Drosophila is small and the female Drosophila is large. Like a fruit fly, male fruit fly is small and the female fruit fly is large. So with the use of easy observation, we segregating the male and female fruit flies. So within a single reproduction, a large number of progeny are produced. Within a single reproduction, a large number of progeny are produced. Large number of progeny means one time reproduce the particular Drosophila reproduce means it produce more number of progeny. We analyze the various characters in the progeny with the use of single fruit fly. And another point, for taking the fruit fly for his research, the male and female is easily distinguishable. Earlier told. Male is small and the female is large. So with the use of easy observation, we differentiate the male and fruit fly and female fruit fly. So variation with low power of microscope also we see. Normally you move into the chromosomes or genes means we need a high power of microscopes like electron microscopes. But in morphological changes of the particular fruit fly, with the use of simple microscope, we observe the changes in the progeny. For example, due to mutation, example, a brown colored eye formed in the progeny means with the use of simple observation, we conclude, okay, that is actually a brown colored eye is a new variation, but it not present in the parents. So with the use of simple microscope, simple lens, we conclude, we pointed out the variations in the progeny. So with the use of these advantageous characters, particular T.H. Morgan is selecting the Drosophila melanogaster for his research. So what research? The chromosomal theory of inheritance. For experimentally proving the chromosomal theory, T.H. Morgan selecting the organism Drosophila melanogaster for proof. For normally in science we need proof. In any aspects we need proof. Normally we know the plants performs photosynthesis and the glucose are produced and glucose is lastly converted into starch. With the use of iodine test only we confirm starch is present in the leaf. Then only we accept during photosynthesis plant prepares starch. Okay, with the use of research only, with the use of result of the particular research only, we conclude, normally we like a scientist conclude the particular research and the particular chromosomes only inheriting the substance or hereditary material of the organisms. So we need evidence, then only the scientific theory is accepted by other scientists. For scientific theory, for experimentally verifying the chromosomal theory of inheritance, the T.H. Morgan selecting the Drosophila melanogaster organism. For selecting the T.H. Morgan, these are the advantageous characters of Drosophila. These characters, it is very useful for this research. So the T.H. Morgan selecting these organisms for this research. Okay. Then the T.H. Morgan research in the next section. Yes. Then experimental verification of chromosomal theory of inheritance. Experimental verification is nothing but how experimentally T.H. Morgan prove chromosomes only hereditary substance. Chromosome only inheriting the character from one generation to next generation. Normally T.H. Morgan take selecting Drosophila melanogaster for his research. In last video we learned why T.H. Morgan selecting the Drosophila melanogaster. Now with the use of research, how T.H. Morgan proved a chromosomal theory of inheritance with the use of linkage and recombination. Now we learn linkage and recombination. Linkage means what? The close association between the genes in a chromosome. Normally genes, for example, human contain around 30,000 genes in 46 chromosomes. So each chromosomes contain various numbers of genes. Normally chromosomes, inside the chromosomes, genes are closely located or maybe loosely located. So linkage is nothing but how genes are linked or closely associated within the chromosome. Listen, this is structure of chromosomes. All are structure of chromosome. Already I have studied in 11th standard. Is actually the shading part is nothing but genes. For example, Y plus W plus gene are present in a chromosome. Here Y W genes are present in a chromosome. So actually this is chromosome. All are chromosomes. Here also all are chromosomes. With the use of chromosomal linkage or gene linkage, the particular T.H. Morgan proved a chromosomal theory of inheritance. Normally in garden pea plant, the Mendel's research is correct. It shows the 9:3:3:1 ratio in dihybrid cross. But the same cross done with the use of Drosophila, that is fruit fly, by the T.H. Morgan. But the result is deviated from the Mendel's result. According to the Mendel's result is 9:3:3:1 only, it is expressed in F2 phenotypic character. But in T.H. Morgan's research in Drosophila melanogaster, the fruit fly organism, the result is deviated. Result is not exactly same to the Mendel's result. So how the results is deviated? The T.H. Morgan performed various research how the result is deviated from the Mendel's result. So one of the research of the T.H. Morgan is, so normally T.H. Morgan is proving for need to prove a chromosomal theory with the use of Drosophila melanogaster, but the research result is actually similar to the Mendel's result, but it is deviated. It is not in the similar to the Mendel's result. The result is deviated from the Mendel's result. So how the deviation occurs? Then only he performed the research with the use of Drosophila melanogaster. Lastly he explained, T.H. Morgan explained that due to close association of gene in a chromosome, only it modify the result. So he formulated another two terms from his experiment, that is linkage and recombination. Linkage is nothing but the close association of genes in a chromosome. Recombination is nothing but that is variation. How the organism is different from the parent organism, that is recombination or variation. The linkage means close association between the genes in a chromosome. A recombination means a variation in a characters, variation in a progeny, that is recombination. So normally the Mendel particular T.H. Morgan selecting the Drosophila with the use of two characters. He take yellow bodied, white eyed female. On Drosophila we know Drosophila fruit fly is female is larger and male is smaller. So we take a particular he take a yellow bodied white eyed female. Yellow, the structure of him and particular Drosophila melanogaster is actually yellow surfaced, yellow bodied plus white eye female crossed with brown bodied red eyed female. Brown bodied red eyed female. We take two organisms, one is female, one is male. The female character is yellow bodied white eye and the male character is small. Male character is small, male actually is small. The character of male is brown bodied red eyed. He copulating, he mating the male and female. The female character is yellow bodied brown eye. It is not brown bodied, it is actually a brown eye. That is brown eye. Okay, you take Drosophila, female yellow bodied white eye and the male is brown bodied yellow eye. Correct only, brown bodied only. Okay, you take yellow bodied white eye female of Drosophila melanogaster plus brown bodied red eye male. He mating the male and female and progenies are produced, but the result is not like Mendel's result. Actually, we know according to the Mendel's result which character is expressed in F1 generation. Only the yellow bodied white eye only it expressed due to dominance, but here is different characters are produced, but is deviated from the Mendel's result. Is not shows 9:3:3:1 because we taking two characters, yellow bodied white eye. So two characters, that is actually a dihybrid class. The T.H. Morgan also performed the same dihybrid class but in Drosophila melanogaster organism, not in pea plant. In pea plant Mendel's performed dihybrid class in pea plant, but in T.H. Morgan performed same dihybrid class in Drosophila melanogaster, yellow bodied white eye female crossed with brown bodied red eye male, but the result is deviated from the Mendel's result. How result is come? Listen the drawing, listen this graph. Actually this is a parents, is actually the this is actually a female parent. Female parented what character? Yellow bodied white eye, that is a female character. Here yellow bodied means capital Y or maybe small y, this is white. In this class we take yellow bodied Y and white bodied W. So yellow bodied character is controlled by one gene, we know, and white eye also character controlled by one gene. That is actually in the rose color is nothing but gene. So actually this is chromosome. Chromosomes, in the chromosomes only genes are present, we know. Chromosomes only genes are present. In the gene is nothing but yellow bodied, the character is controlled by this gene and the white eye character is controlled by this gene. Here also yellow bodied controlled by this gene, here also white eyed controlled by this gene. Actually this is two chromosomes, is actually the chromosome is XX. Normally we know X chromosome is a sex chromosome in female and 2X are present in female. In male means sex chromosome is X and Y. Okay, this is actually X and Y. This is X chromosome, this is Y chromosome. So you take yellow bodied white eyed female and brown bodied red eyed. If a brown bodied, it is actually a brown bodied red. Brown bodied we know brown bodied, but in genes in Y plus means it is brown bodied gene and W plus means it is red eye gene. Okay, you take 2X chromosome in the female, yellow bodied white eye, and the male means white eye, yellow bodied white eye and brown bodied red eye. Here brown bodied red eye. The Mendel like T.H. Morgan selecting yellow bodied white eye female crossed with brown bodied red eye male. This actually is represent female 2X chromosome and it is represents male XY chromosomes. Okay, that is actually a parent. In generation, F1 generation, in F1 generation the progenies are produced. How progenies are produced? In X chromosome from the female is mate with X chromosome in the male. This is actually XX. Normally in hybridization, what occurs in the reproduction? X chromosome from the female and X chromosome from the male combine to form one progeny. This is actually X chromosome, So actually what, female or male? That is female. Female is represent the symbol. So one male X chromosome, that is a female X chromosome, and the male X chromosome join and form another one progeny, that is XX chromosome, which contain Y plus YW and Y plus W. The genes are yellow bodied white eye gene and brown bodied red eye gene in a one individual. Then here in the X, the second X is crossed with Y, X and Y. This is X actually, this one 2, this join to form this organism, YW, and here is actually genes are in the same generation. This is actually Y chromosome. Actually what is this? This is male. Okay, normal sexual reproduction only. So one female and mate with the male, one female sex chromosome mate with the male sex chromosome to form one progeny. Another one female sex chromosome mate with the male XY chromosome form another one progeny. This is done actually F1 generation. Okay. This is actually F1 generation. Then with the use of F1 generation, he hybridizes to form a F2 generation, same like dihybrid cross performed by the Mendel. How? Listen. In actually in F1, 1 and 2 organisms are produced. In F2, totally 8 organisms are produced. First 4 are the same, next 4 also same like first 4. So I explain only 4 progenies in the F2 generation. In F2 generation, listen, the same in F1 generation, listen, the X and here also is actually X, here Y, because male only XY chromosome only sex chromosome. The particular X mated with in the X the chromosome to form an own progeny. It's actually this one, XX. And here X chromosome and mate with the Y chromosome, here X and Y, and Y plus and YW. This is actually one wild type and white and yellow white and yellow. So normally we in first section itself I told this actually a result is deviated from the Mendel's result. So only 2 characters are expressed according to the Mendel's law of inheritance, but in T.H. Morgan research the F2 generation is not like the parents, is totally deviated. This is wild type is not present in the parents. Even white also, yellow, white and yellow also not present in the parents. So in this research, the F2 generation shows various result is deviated from the Mendel's result. So in this result, listen, here this is actually a new character. Listen, half of the chromosome or 80% of the chromosome is a parent chromosome, but this chromosome is different. Here also different. This chromosome also different. Here also difference. So this is the same. It is actually same like parents. Wild type also same like parents. The 1, 2, 3, 4, 5, 6 chromosomes is same like parents. But in one chromosome, 70 to 80% of the 90%, even 90% of the chromosome is similar to the parent. This side similar to the parent. Here also this side is similar to the parent. Only a part of the chromosome, a small segment of the chromosome is deviated, is entirely different. Why? Means because of recombination. Recombination means what? That is variation. Due to this variation, the young one and in F2 generation is different from the parents. So why this is difference means the particular T.H. Morgan explained due to close association of genes. Normally genes are located in a chromosome, we know. The genes are located closely, linked closely. linked closely means it shows only less variation. Actually this variation is 1 point something percentage only, 1 point something, I think that is actually 1 point something percentage, 1 point something percentage only. Listen to the variation in only 1.3 percentage, 1.3 percentage, here 1.3 percentage. Okay, 1.3 percentage variation only occurs. Yeah, this is 1.3 percentage, here also 1.3 percentage, but remaining all are same, here also 1.3 percentage variation, remaining our 99 percentage is similar to the parents. So T.H. Morgan lastly explains the closely linked genes. The genes in a chromosomes are closely linked. Closely linked means it shows less recombination, less variation, only 1.3 percentage. In the next class I show how the loosely linked genes show more variation. According to this view, according to this chromosomal theory, in experimental verification of chromosomal theory, T.H. Morgan explained the Mendel's result is not same like garden pea plant. That is, the result is deviated from the Mendel result, dihybrid result. Due to this result, only 1% of the variation occurs. Lastly, he explained genes are closely linked in a chromosome means they shows less variation, that is 1.3 percentage. So loosely linked genes show more variation. So linkage is nothing but, suppose the genes are located closely linked genes in a chromosome which shows only less variation. Suppose in next section loosely linked genes show more variation. This is actually cross A only. Okay. Listen, this is actually a chromosome. There is XX chromosome, there is XY chromosome, and in the various characters are expressed in F2 generation. It is not like Mendel's result, it is deviated from the Mendel's result. Due to this cross A, T.H. Morgan explained only 1%, approximately 1.3 percentage of characters are newly expressed. At last, he proved the genes are closely linked, closely located in a chromosome means the variation is less. That is, variation means here a recombination. So closely linked genes so less variation, that is 1.3 percentage. But in next class, how the genes are not tightly linked, how loosely linked genes shows more variation, more recombination. Okay. In this class, the T.H. Morgan explains due to close association of genes in a chromosome, it shows less recombination. Okay, then the linkage and recombination. Okay, yes.